Welcome to Thomas Thoughts. In my previous videos, I've had a little bit of difficulty, mainly with delivery of content. And that takes the form of me just being a little bit too vague. I definitely want to go into more detail, and I ended up shooting myself in the foot. I wanted to go into detail, and I kind of rushed it a little bit. And because of that, I ended up not giving out thorough information, or information that would be, in my eyes at least, the full picture. And that's really what I aim to do. So instead of being vague, I'm changing it up a little bit. First off, if you've noticed, if you've seen my other videos, the background's changed a little bit. That's great because now there's no glare. Second to that, I'm going to kind of go back, and for today's episode, I'm actually going to go over a seminar that I attended at university. And the reason I'm going to do this is for two reasons. Number one, it is extremely fascinating. And number two, it'll help me kind of digest the information a little bit further so that I have a better understanding. Because going over notes is, one really great way that you could study. So without further ado, let's dive in. You need a little bit of basis in genetics. Now there's a central dogma to this. The central dogma is you have DNA, which codes for RNA, which codes for protein. And that's the central dogma of biology. That's kind of how the whole thing works. You have your DNA, that codes for all of your proteins. All of your cells that contain DNA have the same DNA. Red blood cells are one of the cells that don't contain DNA, as an example. But the cells that do contain DNA, the reason they do is so that they can create proteins. Now, what's really interesting, and this is kind of sidetracked from the seminar, but what's really interesting is when you take a cell, let's say you take a skin cell, or what's commonly a type of skin cell, rather, called a fibroblast. What they've been able to do in the lab is take the fibroblast and modify its cellular contents. So all of your cells share the same DNA. If you take a cell and put in specific enzymes and constituents that are associated with, let's say, a pluripotent stem cell. It's a really good example. A pluripotent stem cell is a cell that can turn into anything in your body. It's what they use when they're trying to rebuild your cartilage and you're getting stem cell injections. Really fascinating stuff there too. But if you take a fibroblast and you inject it with the contents of what would be in a pluripotent stem cell, the cell functionally changes into a pluripotent stem cell. And that is truly crazy. That is fascinating. So we can actually manipulate any kind of cell. And that's actually how they do it in cultures now. Because earlier in time, we would have to culture, or we would have to take cells from another person and have it be compatible and then have that implanted and stuff like that. And we obviously still do that for the bigger stuff like liver and kidneys. But when it comes to lower impact stuff, so maybe like skin grafts or anything like that, to avoid any kind of immuno response from your body targeting that foreign material, what they do is they actually take your cells and modify them and then grow them in culture so that they are effectively your cells with your DNA and therefore do not elicit an autoimmune response. So that's actually really fascinating stuff. Anyway, more on the seminar. So the central dogma, DNA to RNA to a protein. Now DNA is the double-stranded. It doesn't change. It's static. I mean, it's dynamic in the sense that gene expression, right? It can change and it can shift and it can express different things. But it's static in the sense that you have your DNA. That is your DNA. RNA does not work that way. RNA can be chopped up, RNA can be folded, and all this other stuff. DNA is the code. That is what codes for everything in your body. So when we're looking at RNA, which is the central topic here, mainly, what we have is exons and introns. Once the RNA has been coded for in the DNA. So you have proteins called polymerase proteins, largely polymerase 3 in humans, and it goes through bzzz, and out comes RNA. This particular one is an mRNA, first one that comes off. And you have a long segment of it. Now, you have exons and you have introns. And the introns have commonly been known to be spliced out. And they are. By and large, they're spliced out. As far as we know, that's where we're at. But this seminar kind of defied that logic a little bit, because we were talking about the regulation of gene expression of mRNA. So here, only a small percentage of a gene is actually an exon. So after it's all spliced and put together, you have your gene. Now, what that codes for then is for another protein, or a ribosome, which is a macromolecule. It's a protein-ish thing. It's huge. And it comes in and it locks in other RNAs called tRNAs, which hold on to amino acids. Amino acids code for the protein. Or not code for the protein. Amino acids are the protein. The protein is composed of amino acids. So you have a protein, has a whole bunch of amino acids. For it to get to that point, amino acids have to come in and plop themselves down in sequence, and you get another chain. So basically we're going from chain to chain to chain to fold. So, and the fold there, that's the protein. So splicing has to be incredibly tightly regulated by your body, because there's a lot more introns than there are exons. So if your body isn't on par with that, it's going to splice out introns and maybe even part of an exon. And if it does that, then you get the wrong protein. So it has to be extremely tightly regulated. And what we end up with is something called alternative splicing. This is the ability to get different gene products from the same original mRNA or pre-mRNA, which is fascinating. So again, you create mRNA. They call this pre-mRNA. Before it's spliced out, you have your pre-mRNA. Now, depending on what segments are spliced out at that time to make it shorter, you get a different gene product, which means you get a different protein. And this allows for huge levels of regulation. Now, a really, really important concept in biology is something called feedback inhibition. This is a concept where the products of a sequence of chemical reactions goes back and inhibits any more of the product being created. This is incredibly important. So when we're talking about genes, what you end up with is gene products ceasing the construction of new proteins. Or not gene products. Chemicals in your body going back and stopping the expression of certain genes to create proteins. Now, this can change in response to stress, nutrient depletion. Are you getting enough sleep? Are you getting enough sun? Are you meditating? Are you exercising? All of these things have bearing over your genes. Now, understand, you have a lot of DNA, relatively speaking. So a lot of it is turned off and a lot of it is turned on. And that has to do with whether or not you're eating healthy, you're getting sun, the things I mentioned before. If you're not doing those things, certain genes are not going to be expressed. And when certain genes are not expressed, certain proteins are not expressed. Here's a level deeper, okay? If you're not doing these things, proteins are expressed too much. Proteins are expressed too little. So it's not a matter of just on or off. There's a level of optimum that all humans can reach within their bodies, assuming functionality. We can all reach that optimum level. To do that, our genes have to be functioning properly. So if you're not doing these objectively healthy things, what you end up with is dysregulation of proteins and enzymes. What that can lead to is certain things not being cleared out of your body, certain things not being made, certain cell walls not being as stable as they should be, okay? So all of that ties in to your experience through your life. That all of that ties in. So if you have brain fog frequently, or have trouble with your memory. Let's say that you're right around my age. You're like in your 20s, mid 20s or something, and you're having brain fog, you're just lethargic. It is incredibly likely that it has a lot to do with how you're treating your body. And hopefully that's not like new news to anybody. You know, I mean, I would hope not, right? But the case is, yes, these systems are incredibly complex. But at the same time, most of the complexity is handled by your body, which has had millions of years to develop its mechanisms. All you've got to do is give it what it needs to function optimally. Case in point. So, mammalians have something really interesting that we can do, and it's called intron retention. Now, earlier I mentioned that introns are spliced out, and then you have a collection of exons, which are then used to create protein. Here's what she was talking about. Introns have signaling pathways on them that, if they're read by a protein, will feedback, remember, feedback, inhibition, but there's like a whole bunch of different feedbacks. It feeds back, and you create a different protein based on the environment. So all of these things tie in. It's just complex but fascinating. So pre-mRNA splicing takes place while the strand is being transcribed from DNA. So while that mRNA strand is still coming off the DNA, and you're getting your string, that is right there being sliced up, and you're getting your more complete product. So how are the introns recognized and removed? Well, the histones, so I didn't mention that earlier. Histones are the protein functional units that DNA wraps itself around. Okay, so you have DNA, which is wrapped around histones, which bunches together, and then you get chromosomes. That's really the fundamental. That's like chromosomes, and then the whole thing wraps around. It's called supercoiling. Really crazy. So histones undergo changes to their structure. So you have the protein, and oftentimes, most times, pretty much, they'll have a little piece hanging out from them. Now, depending on whether or not that little piece of the protein has a certain signaling group on them, notably acetylation versus methylation, those are the two. As far as I know, I mean, I'm still learning this stuff, so keep that in mind as well. But you have acetylation and methylation, and so depending on which one of these is on the histone, it's going to change the expression of the gene. So there's a couple different histones, and I don't want to really get into the more complex parts of it because it's really not super necessary, I suppose. But you methylate a histone, and what you end up with is a different expression of the gene that's attached to it. So there are all of these different proteins that end up methylating and demethylating and acetylating and deacetylating. Now, hold on, I'm reading my notes. It's from the source, best of my note-taking abilities, to you. So what can I say? Okay. On my paper, I have not a list, but I have a bunch of these proteins. PRP45, EAF3, SET2, PRP19, EIF3, I already said that, NTC. So the point is, there's a lot that goes into it. It's not just as simple as boom, 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 protein, boom, body, right? I mean, we could have guessed that, right? We could have guessed that. But the bottom line is there's a lot that goes into it, which is just so crazy, because how this relates to our lives in the big picture is that if I guess, to put it simply, most things that you're experiencing can be manipulated. Now, I'm not talking about psychology here, okay? Personality, all that stuff, that's another level. Not necessarily deeper or more superficial, it's just another level. I guess level implies that. Another domain is a better word. So psychology is another domain. Now there's a lot to that as well, but as it pertains to just your biological functioning, there is a really complex series of events that has to occur. Now, all of these proteins that I mentioned work together to sense your environment, sense the environment that you're in. And when they sense the environment that you're in, and that will change which genes are expressed, which ones aren't, which proteins are created. Now, it's important here to remember once again that proteins are where the action is. DNA is vital. It's vitally important, but you wouldn't be a functioning organism without your proteins. And your proteins, the highly functional ones or the most optimal functioning ones in your body, wouldn't be possible or are currently not possible unless you're treating your body as a separate entity, if you will, okay? Now bear with me. If you treat your body not as just your meat sack that you walk in or do whatever, but instead as an organism that you're caring for, you can alter many facets of your life. Because everything that you put in your body is used to perpetuate your body. Same thing with brain cells. Without diving into psychology, right? Because obviously neural connections and all that stuff and firing and neurotransmitters, which by the way, obviously neurotransmitters are also created by the things you put in your body. But the bottom line is, here's a good example. Fish oil. Fish oil contains EPA and DHA. Both of those are really important for your brain, because your brain takes DHA and integrates it into the membranes of your neurons. You don't have quality DHA, you don't have quality neurons. It works that way for everything. Everything. So, back to the complexity here. Yeast. Yeast has a protein called GCR1. GCR1 regulates metabolic programming as cells grow to saturation, experience nutrient limitation, arrest growth, and enter the stationary phase. So, yeast can go into something called a diaxic shift. I think I'm pronouncing that right. Now, this allows the yeast to live for a long time without utilizing its metabolism, but stay alive. So that's how you can dig up a grave from like hundreds of years ago and pull it out and open it, and if you analyze it, you'll find dormant yeast cells living there with no real organic material to thrive on, because they can put themselves in a stasis. And they do that through this gene called GCR1, because what happens is GCR1 is able to sense its environment. So I guess GCR1 is a really great example on how to kind of bring this whole thing full circle. GCR1 is able to sense the environment of the yeast and realize, okay, I'm not getting enough of this nutrient that's necessary for X. Okay, I don't have enough of boom, 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 boom. And what you end up with is an activation of certain genes that allow the yeast cell to go into a stasis mode and effectively almost turn off its metabolism. It's at a very low rate, so it can survive for a very, very long time. This is incredible. This is incredible that yeast can do that. So yeast are yeast, but as humans, we can do that. Not go into stasis, not cuddle up in a hole and survive for hundreds of years. No. But based on how we treat our bodies, we change the way that our bodies operate. And that's a fundamental fact. It's fundamental. So next time you catch yourself thinking, I'm a little bit down, or maybe a lot down, or man, I just feel so lethargic and tired, or man, I just don't want to do that. Of course, these are all negative things. So generally it's negative. You see yourself gaining weight, losing interest in all of these things, maybe not being able to sleep. I urge you to look at your lifestyle. And this is not new. This is not new. But I urge you to just take a look at it. And also realize something really important. And I mentioned this in my past videos. It's that if you are struggling with the discipline to eat healthy, remember a few things. Number one, it is likely not you. It is your microbiome telling you to eat poorly because that's what it thrives on. And two, I forgot to. Because number one is enough. Understand, in order to change your eating habits, you have to change your eating habits. You just got to do it. And once you do that, after like 3 weeks of really kind of putting effort in and cutting out sugar, also if you're really in the trenches, you're probably going to go through sugar withdrawals. Get through it. You'll come out on the other side of the rainbow. And everything will start to regulate itself a little bit better than it did before. And hopefully, you'll clear up that down cycle. You will start losing weight if you're overweight and you're eating healthier. Almost guaranteed. Of course, there's caveats. And you will likely get more motivation because you'll feel better. Disclaimer. If you are in a down cycle and you are eating healthy and you are doing all the right things and you are still really down, seek medical attention. Don't let that go unchecked, because you're the only you're ever going to get. So take care of yourself. This has been Thomas Thoughts, and I will see you again.